Here is the background of Operon. The Operon is a genetic regulatory system found in bacteria, such as E. coli, that controls the expression of genes involved in the metabolism of the sugar galactose. The operon comprise a cluster of genes which encode the enzymes necessary for the conversion and utilization of galactose through an amphibotic metabolic pathway. The expression of these genes is tightly regulated by a complex interplay of regulatory proteins and DNA binding elements, including repressor proteins. These intricate regulatory systems allows bacterial cells to efficiently utilize galactose as a nutrient source when preferred sugars are not available, while conserving energy when other sugars are present. This is the structure of the gall operon. Gall operon contains two operators which are OE and OI. OE located upstream of the promoter while OI located in the structural gene region downstream of the promoters. The operon also has two overlapping promoters, P1 and P2. P1 responsible for high-level expression when no sugar is present, while P2 drives low-level expression in the presence of other sugars. Each promoter responds to different regulators for coping with physiological needs, as enzymes encoded in the gall operon are needed for both catabolic and anabolic metabolisms. Then, gall operon also includes four structural genes, which are GAL-E, GAL-T, GAL-K, and GAL-M that encode enzymes for galactose metabolism. It also contains binding sites for the DNA binding protein HU, which is called as HBS. This site allows for the binding of the DNA. At the same time, it also has an activator binding site where CAMP-CRP complex will bind there to activate transcription. Lastly, gall-operon also has repressor protein GAL-R that binds to the operator regions to repress the transcription in the absence of galactose. Next, what it codes for. Structural genes of the gall operon, which are GAL-E, GAL-T, GAL-K, and GAL-M, encodes enzymes necessary for the conversion of galactose. GAL-E encodes UDP glucose 4 isomerase and enzymes that interconverts UDP glucose and UDP galactose. GAL-T encodes galactose 1-phosphate uridyl transferase and enzymes that converts galactose 1-phosphate into glucose 1-phosphate. Then, GAL-K encodes galactokinase and enzymes that phosphorylates galactose to galactose 1-phosphate. Lastly, GAL-M, which is galactose mutaroatase, will catalyze the conversion of beta-D-galactose into alpha-D-galactose. These enzymes encoded by the structural genes help channel galactose into the LELWA pathway, which is the primary metabolic pathway for galactose utilization in bacteria.
Next, let's look at the negative regulation mechanism of gall operon. Before that, let's examine the condition. So, the repression of the gall operon happens in the absence of galactose and it is subject to catabolic repression in the presence of glucose. Catabolic repression means that when there is a high level of sugar, which is glucose, present in the environment, the expression of the genes in the gall operon is repressed or turned off, even if the galactose is present. Galactose acts as an inducer of the gall operon by binding to the GAL-R repressor, causing a conformational change that releases it from the operator. Without the presence of galactose, the GAL-R repressor remains bound to the operator, maintaining the operon in a repressed state. Now, let's go through the mechanism together. The negative regulation mechanism of the gall operon of E. coli is regulated by the gall repressosome, a high-order nucleoprotein complex that involves the interaction between GAL-ROE and GAL-ROI complexes. In the total absence of D-galactose, the gall repressor GAL-R will recognize and bind to the DNA operator sites, which are OE and OI, forming a tetrameric repressor in the regions of the P1 and P2 promoters. A histone-like protein, HU, assists the GAL-ROE complex and the GAL-ROI complex in stabilizing a higher-order complex structure of repressosome, which results in a DNA loop. Synergistic binding of GAL-R to distal sites forms a 113BP DNA loop, which is a topologically closed domain containing the two promoters P1 and P2. The DNA loop is bound at the histone binding site HBS, which is located at the apex of the loop where HU binds. This GAL-HU-mediated DNA looping represses both the P1 and P2 promoters of the GAL operon simultaneously. The closed DNA loop either prevents the unwinding of DNA needed for open complex formation or impedes the processive DNA contacts by RNA polymerase during transcription initiation, thereby repressing both promoters. As a result, structural genes are not transcribed.
Moving on to positive regulation mechanism. The activation of GAL operon transcription expression is mainly regulated by contact activation with GAL-R and the role of CAMP-CRP complex (CCC) and its interaction with RNA polymerase. Referring to the picture shown, in the total absence of D-galactose, both P1 and P2 are repressed by the repressosome through formation of DNA loop as explained earlier. Low concentration of intracellular galactose allow GAL transcription at a low rate. This condition will contribute to disassembly of the repressosome with dissociation of GAL-R from operator OI without dissociation of the GAL-ROE complex. This only allows P2 transcription and very little of P1 transcription because P2 drives low-level expression in the presence of other sugars. But at condition where high concentration of extracellular D-galactose, GAL-R will start to dissociate with operator OE, freeing the DNA from GAL-R. This step is mediated by the role of CAMP-CRP complex that further enhance the repression of P2 and activation of P1 transcription to allow expression of the GAL operon for D-galactose catabolism. The role of CAMP-CRP complex. Galactose needs to be broken down by the GAL operon, which is regulated by the CCC. This complex is a regulatory protein that plays a key role in regulating the expression of GAL operon. Low level of glucose will signal the cell to use other alternative sugar to counteract the low intracellular glucose level and low intracellular ATP level. One of the alternative sugars includes galactose, which can be used to be converted to glucose via the lower pathway. Inhibition of glucose metabolism, which is glycolysis, will lead to decreased levels of intracellular ATP. Decreased level of ATP in the absence of glucose and high level of galactose will relieve the inhibition of adenylate cyclase, activating it to convert the remaining ATP to CAMP. High level of CAMP will allow it to bind to CAMP receptor protein, CRP, forming CAMP-CRP complex. The CRP binding to CAMP will cause conformational change that make it capable of binding to specific DNA sequence in the promoter region of the GAL operon. The binding site is called CAMP receptor site, which is located upstream of the GAL operon promoter. This binding enhances the binding affinity of RNA polymerase to the promoter region. The CCC, along with RNA polymerase, forms a stable complex at the promoter. This interaction results in the recruitment and proper positioning of RNA polymerase, facilitating efficient transcription initiation and promoting higher levels of transcription from the GAL operon from P2. The genes in GAL operons such as GAL-E, GAL-T, and GAL-K will be expressed to allow efficient utilization of galactose as an energy source in E. coli.